Hi, my name is Carl Bosque Cordero. I'm a PhD candidate from the University of Puerto Rico, Rio Piedras campus. Currently, I'm working over at Dr. Carlos Jimenez Neurophysiology Laboratory over at the Medical Sciences campus. The title of my project is IH blocker reduces cocaine-induced firing patterns on putative dopaminergic neurons of the ventral tegmental area. Our laboratory explores and studies the effect of cocaine on the brain, specifically on the reward pathway, which is an interconnected region that regulates pleasure, reward, and motivation. Within this pathway, we evaluate dopaminergic neurons of the ventral tegmental area. These neurons are implicated in reward related behaviors. We also study H current, which is called the hyperpolarization activated Cadmium current. It's a mixed current carried by both sodium and potassium. It's widely distributed in excitable cells, such as dopaminergic neurons. It's a depolarizing current that affects cell excitability by generating pacemaking activity, determining the resting potential, and firing frequency. It also modulates the amplitude and duration of hyperpolarizing inputs. Our laboratory has previously demonstrated that after cocaine sensitization, a chronic cocaine model, there's a significant reduction of IH by 40%. One of the current hypotheses of this reduction is that it serves as a homeostatic regulation to oppose cocaine enhanced excitability and regulate firing activity. To explore this hypothesis, we need to evaluate in vivo firing characteristics, mainly by using in vivo anesthetized single unit extracellular recordings, where we will have the whole brain configuration and we can measure specifically bursting activity. A burst is defined as a set of two or more action potential within a short interval of time, followed by an extended period of pause. It's important to denote that phasic activation of dopaminergic neurons of the ventral tegmental area is sufficient to drive behavioral conditioning. So we decided to explore what is the effect of blocking this current on in vivo spontaneous activity. Here we see a naive animal in a stereotax which is anesthetized and using a double barrel pipette, which is composed by a recording electrode and a perfusion pipette containing IH blockers, ZD. We will record spontaneous activity for 100 seconds and we will perfuse the IH blocker and evaluate the effect after this perfusion. Our hypothesis is that IH blockade will reduce firing rate and bursting frequency. Over on the results, we see on figure A, sample traces from the experiments. Each dot denotes bursting formation by the program. We see that after perfusion of IH blocker, there's a significant decrease in firing rate. We also determined that there's a significant reduction in bursting frequency and a significant reduction of percent spike in burst. BTA dopaminergic neurons can be categorized in different firing patterns, mainly on four categories, which are denoted by the mean firing rate and the percent of spikes in burst. Evaluating all these neurons, we didn't demonstrate we didn't find a specific difference within each category. We pulled up all of our data and evaluated the interspike interval histogram, which denotes the time within each spike. And after the perfusion of IH blocker, there's a significant increase in time within each spike. We also found that there's a significant reduction in number of events, which can be demonstrated that there's a reduction in spike formation. We also wanted to explore the effect of acute cocaine on dopaminergic neurons excitability. This is a data from a previous publication from Dr. Megan Creed, where she demonstrated after a single cocaine injection, there's a significant increase after 3, 6, 24 hours, and 5 days of a single cocaine injection. It's also demonstrated that only after 24 hours of this acute cocaine administration, there's a significant increase in bursting activity. We decided to explore the effect of acute cocaine using a novelty model, cocaine novelty model, where we administer a cocaine in a new cage where the animal is exposed for 1 hour. After the cessation of this hour, the animal is placed back to his home cage, and 23 hours we pass this animal to the experimental stereotaxis. So using the same experiment, a double barrel pipette is placed and we record baseline activity and we perfuse IH blocker after 100 seconds and evaluated the effect of IH blocker on cocaine-induced excitability. Our hypothesis is that IH blockade will reduce cocaine-induced firing rate and bursting frequency. So we evaluated using in vitro recording, wholesale patch clamp recordings. We measured IH amplitude 2 hours and 24 hours after a single cocaine injection. We demonstrate here that there's only a significant 25% reduction of this current after 24 hours of acute cocaine and not 2 hours. So this demonstrates that this reduction is a progressive reduction after acute cocaine administration. We also mimicked our previous experiments after 7 days of chronic cocaine model, and we also demonstrated revalue the 40% reduction of this amplitude. Over on the in vivo recordings, we demonstrate that IH blockade can significantly reduce cocaine-induced firing rate, bursting frequency, and percent of spikes in bursts. Using different firing patterns classification, here we demonstrate that the specific population within this category, the low firing rate and high bursting group, is the mainly affected by ZD perfusion. So we demonstrated within this subgroup using an interspike interval histogram that there's a significant increase within the interspike interval of these spikes. Also, there's a significant reduction on the number of events. So by now, I hope I have convinced you that IH blockade reduces firing and bursting patterns on VTA-DA neurons by increasing the interspike interval. And specifically in the acute cocaine group, the low firing, high bursting VTA-DA cells, IH blockade diminish acute cocaine novelty, firing and bursting patterns, also by increasing the interspike interval, and that IH reduction after chronic cocaine could serve as a homeostatic modulator to oppose cocaine-induced excitability. So I would like to acknowledge my laboratory, Dr. Carlos Jimenez Rivera Neurophysiology Laboratory, over at the dopamine and neuronal assemblies teams over at the University of Bordeaux, specifically Dr. Georges and Dr. Foyce, and the NSF PIRE program and the RISE program that helped me with all the funding and all the help for all these experiments. I'll be glad to answer any questions you have for me. Thank you.